All right, so we left off with the Lac Operon, which is our inducible operon. All right, the Lac Operon is normally off. You need to have lactose present in order to turn it on. Okay, the other type of repressors that we've got in bacteria are repressible operons. And in terms of that, the Tryptophan is a great example of a repressible operon. The Tryp stands for tryptophan. Tryptophan is one of the 20 amino acids. The bacteria wants to make that product. Unless there's such an abundance of it, it doesn't need to. So what happens with the Tryptophan, normally the repressor is made, but the repressor is inactive. All right? And because the repressor is inactive, it's not in the right conformational shape to bind to the operator, which means our RNA polymerase can come in. We'll make that at that oval. Sits down on the promoter, and it starts reading that information within the DNA. Okay? Then what happens? You get transcription of the genes needed for tryptophan. And I know in your textbook and in the PowerPoints there are different letter abbreviations for the genes that are involved in tryptophan. I'm really not concerned that you know what all the genes to make the amino acid for tryptophan are. All right? Just know genes for tryptophan, there's more than one gene product that's made. All right? So in terms of that, what we end up having happen, we've got transcription that's going to happen. We're going to make our RNA molecule. All right? That RNA molecule then is going to be translated by our ribosome, and that's going to make all of our tryptophan protein. All right? So the box is here. This is our tryptophan protein. Bacterial cell can then use that tryptophan to make other proteins, can use it for other sources within the bacterial cell. But in terms of that, this is one pathway you typically want to have on. All right? The only time you would want to turn this off, all right, is if our levels of tryptophan get high. Oh, this is a squeaky pen. Sorry about that. Let me see if I can get one that's less squeaky. So we've got high levels of our tryptophan. All right? What that does now, our tryptophan can interact with our repressor protein. And what that's going to do to our repressor protein, okay, this is our repressor. This now, we'll put this over at the side here. This is our tryptophan. It's going to result in a conformational change on the repressor. Now what can happen? That repressor is the right shape, in the right form, and it's going to now, sorry as I erase this. Good thing their bacterial cells and our cells are faster at this than I am. All right? What's going to happen now, that repressor can sit on our operator region, and now that RNA polymerase can't bind to the promoter region. All right? And it can't transcribe the genes. So when the repressor sits on the operator, what that's going to do now oops, spelling. It's going to switch it to the off position. All right? So whether we're talking about the Lac Operon or the Tryptophan, it's off when the repressor is bound to the operator. What results in the repressor binding to the operator is a little bit different with the repressible system, because you've got to have lots of tryptophan present in order to get the repressor in the right conformation. With our Lac Operon, the repressor there, remember when lactose bound to the repressor, it changed the conformation so the repressor fell off the operon. Okay? So our Lac Operon is normally off. The repressor falling off results in transcription happening and turning on that gene. In the case of our Tryptophan, normally on. When you've got high levels of tryptophan, that'll trigger the repressor to bind, turn it off. So this one's usually on. You need the repressor to turn it off. Okay? Lac Operon, normally off. You need the repressor to fall away in order to turn it on. That's really the difference between something that's inducible versus repressible. All right? If you have any questions, let me know. Thank you for your attention.